Everyone. I am Isabel Lusada, and I'm the CEO and founder of the Amyloidosis Research Consortium. Welcome to our ARC Talk series, and I'm delighted that we have Dr. John Berk, who is joining us today, and he's Professor of Medicine and Assistant Director at Boston University School of Medicine and the Amyloidosis Center at Boston Medical Center. Dr. Berk has amazing expertise in amyloidosis. I'm sure many of you will know him, have heard him speak, have met him, or see him at regular intervals. And he has a real emphasis on ATTR and localized forms of amyloidosis. ARC Talks is brought to you by the Amyloidosis Research Consortium. We're a non-profit, and our vision is to make a significant impact on the curability of amyloidosis. We have 4 areas of focus: improving the speed and accuracy of diagnosis, increasing the understanding of the science, accelerating regulatory approval and reimbursement of effective treatments for patients, and enhancing care and quality of life for patients and caregivers throughout their journey with amyloidosis. I'd like to thank the sponsors for today's meeting. There are over 7,000 rare diseases, and less than 5 have a treatment. In ATTR, we've been really fortunate, and in the past 5 years, 4 treatments have been approved. And there are ongoing clinical trials studying other novel treatments. So the treatment landscape is truly evolving, and I'm really delighted to welcome Dr. Berk to walk us through where we are today and future directions. I'll hand over to you. Thank you.
Isabel, thank you very much. Let's see if I can conquer things here. All right. Can you see my screen?
Not yet.
Not yet. That's interesting.
And while you're looking for it, I just want to remind people that we will, when Dr. Berk presents, we will, after his presentation, we'll go to question and answer. Please add those to the Q&A box, and then I'll moderate those. And try and stay away from personal questions. It's much easier to address general questions. You can reach out to ARC with personal questions afterwards. Thank you. Yep, that looks good.
All right, let's see if we can do this right. And I'm going to switch here. All right, you should just see one slide.
Perfect. Yep, you've got it. Thank you.
Here we go. Let's talk about the topic at hand, evolving treatments for ATTR amyloidosis. And these are my disclosures, which should not bias anything that I'm about to say. So the real question is, what's the problem? Well, the misfolding of liver-derived TTR leads to a disease. Okay, so liver-derived is important. And we're talking about a factory, and the factory is the liver in this case. If you've got a bad product in industry, you change the factory. And in this case, some innovative surgeons in Sweden in 1990 transplanted the liver. And the results were seemingly quite positive initially. And in the graph depicted, we see on the vertical axis survival over time. And the graph shows that the people who are in the blue line, that is early V30M, really survived remarkably well. And that, so if you were early onset, this was a great intervention. And early onset meant very little neuropathy. If you were late in the course of V30M, or if you had some other mutation, whether it was early or late, your survival was not nearly as robust. So it became clear that people had progressive cardiomyopathy and neuropathy after liver transplant, if that liver transplant was performed at the time they had amyloid deposits in the nerve or heart. So an alternative approach was needed. And when you talk about developing new approaches in therapy, there has to be some understanding of the biology of the process. And this schematic, I think, captures that. So you go from DNA code for the protein, which is then delivered to the factory in the liver, which produces the protein. And then within the liver, there is assembly of proteins to form a configuration that involves 4 TTR molecules. And we call this the TTR tetramer. And in this state, the protein is very stable. For a protein that is left unincorporated in tetramer, those proteins have the capability of misfolding and forming aggregates, which we know as amyloid. Neuropathy here is going to be our initial consideration for the simple reason that, as you will understand as we talk, neuropathy drives drug development, and you'll see why. 2 men altered the course of TTR disease, and I think we owe them a huge amount of applause. The first is Jeff Kelly, a brilliant physical biochemist at Scripps Research Institute, who recognized the importance of the tetramer and the rate limiting step in amyloid formation of a release of one protein from this tetramer. So in order to get an amyloid process, you had to be able to disrupt this tetramer. And this ribbon diagram depicts 4 proteins that have come together. This is actually 2 sets of 2, the blue and green, the blue and green above, the yellow and red below, and where these 2 dimers interact, the interface is where a hiding place or cleft forms for thyroxine, thyroid hormone. And Jeff demonstrated that when thyroxine is introduced to TTR proteins and with binding in the tetramer cleft, it stabilizes this tetrameric conformation, making it very difficult to disrupt the conformation and release one protein. If unable to release one protein, then there's no potential for amyloid formation. So you might say, well, why don't we give everyone thyroid hormone? The problem is this is a bioactive substance and that it has many effects throughout the body, so that if you're giving people thyroxine, you're going to make them thyroid sick or hyperthyroid, and you're going to exchange one disease for another. The second process that was really instrumental in clinical trial creation was this gentleman. And I have envied his hairstyle for a long time. This is Peter Dick. Peter Dick is one of the preeminent peripheral neurologists in the world and works at the Mayo Clinic in Rochester, Minnesota. Well into his 90s, he remains active in fine-tuning the development of measures of neuropathy that have permitted observation of and demonstration of nerve injury over time. So with the concepts of how to stabilize TTR tetramers combined with the mechanism that allows us to measure neuropathy progression over time, it was game on for clinical trials. The metric that Peter Dick developed causes migraine headaches to many people, and it's the modified neurologic impairment score plus 7 attributes. And it's a scoring system that involves whether blood pressure changes when you stand, whether your heart rate changes when you breathe deeply, um, whether the electricity in your nerves is conducted normally, whether you have normal reflexes. Computer-driven quantitative sensory testing determining the limits of people's ability to detect change in coolness, in heat, pain, in vibration. And then the majority of points went to um, people who had progressive weakness in their motor function, that is their strength. And as the points go up, your neurologic function is going down. This very comprehensive measure really breaks down to something that should be easily understood and really a major component of everyday evaluations for neuropathy, and that is the interaction with a neurologist. The neurologic impairment score that a neurologist applies is just checking your reflexes, seeing if you can feel light touch, vibration, some coolness, and exactly how well preserved your muscles are. This is a standard evaluation. And as you will see, neurologic impairment score would validate the approval of all of the drugs that the FDA has approved to date. Notably, in diabetic polyneuropathy, which is the reason that Peter Dick was developing this scoring system, there would be a 2-point change in this score over a 2-year period. Keep that in mind. All right, so what are the therapeutic options here? Well, they're depicted in boxes here. Kinetic stabilizers to stabilize the tetramer. mRNA degradation drugs and antibody-mediated clearance. But we're going to start at the beginning, which is the development of drugs that are thyroxine mimetics. And the importance is that they have the same shape and size as thyroxine or thyroid hormone, and they fit into that cleft on either side of the tetramer, stabilizing this tetramer. These are the results, I think most easily understood from the diflunisal trial, in which we enrolled 130 people and monitored their neurologic function over 24 months. The vertical axis here captures the change in neurologic score over time. And the higher the score, the more nerve injury you have. So what you can see is that people who are in the placebo group untreated had a dramatic rise in their neurologic scores, suffering significant neurologic injury. And keep in mind, if you were a diabetic, untreated, it would only be a 2-point change. This is upwards of 20 points in a 2-year period. So the disease we're talking about is diabetic neuropathy on steroids, that you progress at 10 times the speed of diabetic neuropathy. And that's the reason that drug development really depends on neuropathy, and that's the first stage at which a drug should be evaluated. Simply put, you can demonstrate drug effect in a 12-month period by this study, and a shorter period of time in the future studies that we'll see. This blue line here captures drug effect, and you can see there is a markedly lessened neurologic injury. Although I'll point out at 2 years, in the majority of people, there still was some rise in neurologic score indicating ongoing neurologic injury. So the findings from this trial was the TTR indeed was stabilized by diflunisal. The diflunisal inhibited neurologic disease rather dramatically. And I'd point out that almost a third of people had no neurologic progression over a 2-year period. It preserves or improves quality of life because quality of life and neurologic function go hand in hand, and is effective across mutations, all mutations, gender, severity of disease, and it's affordable. Teresa Coelho, a major investigator in the world and centered in Porto, Portugal, um, evaluated the impact of a competing drug that Jeff Kelly developed called tafamidis. And tafamidis at low dose was studied for neuropathy. And what you see here, again, in the vertical axis is survival over time. And you can see in the untreated population that the median survival was approximately 13 years. That if people underwent transplant early in their disease, that their median survival was extended to about 23 years, a 10-year extension. And then if you look at the early data in that population with tafamidis, it looked like the survival either rivaled or even improved that seen with liver transplant. So what do we know about TTR tetramer stabilizers? They slow the course of disease. They rarely stop disease progression, but it can happen. But is slowing disease enough? And I think the answer to that clearly is no. We don't want to rest having a therapy, a primary therapy that simply slows disease. We want to stop it or reverse it. So what's the next alternative? Well, the next alternative for a variety of reasons turned out to be mRNA degradation drugs. What does this really mean? So the philosophy here was, instead of stabilizing the protein and preserving it, this was the objective of eliminating the amyloidogenic protein. How do you do that? Well, the idea was that if you have the blueprint in the architect's office, there has to be a messenger that takes the blueprint information to the factory where production will occur. And these drugs are designed to eliminate, or as I put it, to shoot the messenger to prevent the delivery of the information to the factory. No information to the factory, no production of that protein, and the building blocks of amyloid then are presumably or theoretically eliminated. There are two candidates here. One is siRNA, which is a biology that won Nobel Award in 2006, and then antisense oligonucleotide treatment. But both are designed to shoot the messenger. So the next heralding we have here is this gentleman, Merrill Benson, a true maverick and a multi-talented gentleman who back in 2006 published this paper documenting the ability to knock out the messenger for production of transthyretin, the TTR protein. And this was done in mice that that were a model for human TTR amyloidosis. A remarkable paper, and he took his findings and simply refused to give up and had multiple negotiations with a very prominent pharmaceutical that was renowned for antisense oligonucleotide work, and that was Ionis. And ultimately convinced Ionis the merit of this approach and that it was worthy of clinical trial. And so Ionis chose to develop inotersen, and a 15-month trial was performed with 172 patients, giving approximately 300 milligrams of the drug by shot to most of the population. And the primary endpoint was whether the neurologic injury worsened or whether the quality of life also worsened. So did the drug actually knock down TTR production? So the vertical axis here is measuring change from baseline. So as you go down, the TTR levels are going down and over time. And what you can see is that with administration of drug, by about 12 weeks, you reach a nadir, which is, we'll say, the mid 70% suppression level. So biologically, the drug is working. And you've seen this graph before. It's the same as the diflunisal trial. We're again looking at increasing neurologic injury over time. And what you can see is at 9 months, there is clear differentiation between those on placebo with worsening neurologic function and those who are better off, less progression of nerve injury, and they're receiving drug. But again, you can see over the 15-month period that there is an increasing median nerve injury by this scoring system. So it very reminiscent of the changes we saw with diflunisal. But here we see that 37% of the population either had stable or improved neurologic function relative to their baseline at entry of study. Inotersen had some safety issues. Over half the people had a significant drop in their platelet counts. There was one unfortunate fatal brain hemorrhage in someone who was unmonitored over time for a variety of reasons. And when monitoring was intensified, no one had platelet drops that were below 50,000. And in order to get brain hemorrhage, you have to have platelet counts less than 10,000. There were some deaths, and there was kidney injury in a very small number of people that probably in part related to the administration of the antisense oligonucleotide. This study was published at the same time the inotersen study was, and this is the patisiran, an siRNA agent. And this was a slightly longer study, some more patients. This was the dose, and the measure was simply neurologic function over time. Did the drug actually drop TTR levels? Again, we see that the answer is yes. And in a subsequent trial for cardiomyopathy, the level was down to approximately the 83 to 85% suppression. So quite effective at dropping it. And you'll see the time between first administration and nadir is not now 12 weeks, but really one infusion, 3 weeks. Okay, this is the same graph we've been looking at. We're looking at neurologic function worsening as we go up, improving as we go down. You see, again, in the placebo group, about a 27 or 28 point increase, that is dramatic neurologic decline over 18 months. And we see there's a marked difference in the people who got drug. But this time, in distinction with inotersen and diflunisal, we see that this is a downtrending graph. What does that mean? That means there's some neurologic improvement, albeit small, a 6-point change, and largely in sensory function, not in the ability to alter strength or to promote walking. But improvement in neurologic function at any level is important. And neurologic and autonomic function is something that improves with this drug as well as with inotersen. Fully 56% of people in this trial improved from baseline. So a really effective drug. Well, is it good for everybody? And is this just good for the people who are really early in the disease, the same way that a liver transplant appeared to be? With one mutation, that is. So what the investigators did after the conclusion of the study was to segregate people into increasing neurologic scores. So worsening neurologic function on the horizontal axis and worsening neurologic function on the vertical axis. And you can see in the pink graph that as the neurologic function is worsening in these four groups, there's acceleration in the nerve injury over time, with the time points being 0, 9, and 18 months as measured in the study. But really striking is the stability of neurologic function and apparent downtrending graph suggesting there's improvement in neurologic function in each of these neurologic groups. So regardless of how severe your neuropathy, this drug works. What it can't do is return the people who are late in the course of disease back to the beginning of normal function. But it can arrest disease and it can cause some sensory improvement regardless of when you start to get the drug. So what's the magnitude of TTR disease? Well, in polyneuropathy, it's guessed, and we really don't know, that there are somewhere between 7,500 and 10,000 people in the United States. And it's estimated 50,000 around the world. And my bet is there's a whole lot more than this. We follow families, many of whom are reluctant to engage with us, or who choose to acquire something like diflunisal through their primary care physician, rather than with interactions with us. Neuropathy, however, is a different kettle of fish. We're not talking about 10,000 or 50,000 in the United States alone with just wild-type disease. It's estimated that there are about 500,000 people. That's a bunch compared to 50,000 for the worldwide neuropathy population. And then when you consider those at risk with V122I, in the African American population, there are estimated to be about 1.6 million people walking around with this genotype. That is not to suggest that everyone with the genotype gets disease, because it's speculated that as low as somewhere between 7 and 20% may be the ones that actually manifest disease, and then everyone else does not. But cardiomyopathy clearly is the prize for pharmaceuticals and their major attention. So what about cardiomyopathy trial? Well, Pfizer gets points for being courageous enough to undertake the first cardiomyopathy trial. And it was a 30-month trial, a little over 400 patients, a couple of doses of the drug tafamidis, and checking, the evaluation was all-cause mortality and the frequency of cardiovascular hospitalizations. So this first graph is looking at the time to first cardiovascular hospitalization. And you can see that over time, in the placebo group, there is a continual and we'll say fairly frequent hospitalization rate that is diminished, so lessened, less frequent hospitalizations in the treatment group, and that the separation of curves, the true drug effect, is notable after 9 months of treatment. So tafamidis reduced hospitalizations by 32%, which is, that's laudable. And then there is all-cause mortality. And in this, we're looking at a survival curve. This is the percent of survival over time. And you can see that the separation of curves occurs after 18 months treatment. And that there was fully a 30% drop in mortality between the placebo and the treated group. But that 30% mortality needs a little bit of further attention. So in the 30-month death rate, almost 43% of the placebo group died, and roughly 30% of those on tafamidis. So being on drug did not completely stop people from dying within a 30-month period. And a 30% mortality rate is rather striking. If you look at 6-minute walk test, the drug did better in preserving the ability to walk, although there's still a decline over a 30-month period. And in the quality of life metric, the Kansas City quality of life, cardiomyopathy quality of life, you can see that the quality of life was better, although downtrending when compared to placebo. So now we've looked at 4 different attributes. One is time to first hospitalization or cardiovascular hospitalizations. The second is all-cause mortality. We've looked at 6-minute walk test. We've looked at quality of life. And all 4 are demonstrate drug effect, but I would emphasize to you that all 4 are still downward trending. So as a primary agent, this is a huge step forward. There is impact on the course of disease, but it is not stopping disease. People progress. Parenthetically, I'd like to just note that the BU diflunisal experience in people with TTR cardiomyopathy is that it prolongs survival. Now, these data are not perfect. This is not from a clinical trial. These are our retrospective review of just our single center experience. But I would point out that Columbia University and Cleveland Clinic Foundation also have similar results. So it suggests that in people who can tolerate diflunisal with cardiomyopathy, and that's not everyone, that it has a survival benefit. We have first and second generation gene silencers. The first generation are patisiran and inotersen, and they've proven their worth and been approved for their impact on neuropathy. And now we have new and improved versions of both, vutrisiran, the new patisiran, and there should be an L in there, eplontersen, which is the new and improved inotersen. Interestingly, the modifications leading to these new drugs are very similar. So 2 very smart companies have developed increased stability of the primary agent and have attached antenna to the front end of the molecules to expedite the delivery of drugs specifically to the liver. That has allowed less drug to be given less frequently. And anytime you do that, there's improved safety and tolerability. So very encouraged just by the design similarities. TTR amyloid polyneuropathy, where do we stand? So we have tafamidis and diflunisal available in the United States. These are the endpoints that were measured, lower leg NIS, and this is total body, so a little bit different measures. The European Medicines Agency conditionally approved low-dose tafamidis for polyneuropathy, obviously in Europe. FDA denied approval in the United States. Diflunisal, we never submitted the data because we didn't have the money or the personnel. What about gene silencers for polyneuropathy? Well, this has been a remarkable run that Isabel already spoke about. For Alnylam, we have Onpattro. For Ionis, we have Tegsedi. And now as a second generation, the first one to reach FDA approval is Amvuttra or vutrisiran from the Helios A study. And we're very optimistic about eplontersen, which is in a phase 3 study as well, has already reported data at a preliminary data point. And what about cardiomyopathy? We have oral stabilizers. We have Vindamax, which has FDA approval, so it has cornered the market. Diflunisal, while it may have benefits, has never been studied in formal clinical trial. AG10 and acoramidis, the 6-minute walk data at 12 months was upended, I would say, by a placebo group that didn't progress at all. for reasons that remain a little mysterious, but is still in clinical trial, and I'm still optimistic about the trial demonstrating drug effect, looking at all-cause mortality and hospitalization rates. And in the gene silencers, we have 3 very important clinical trials ongoing. Patisiran has already reported 12-month, 6-minute walk data that demonstrates drug effect. Vutrisiran is in a 30 to 36-month trial. Eplontersen similarly designed for a 30 to 36-month trial. So stay tuned for the outcomes of those. Well, what about antibody disruption? You know, it was on our diagram to begin with, and there has been interest in this dating back to Mark Pepys, who was knighted for all of his advances and innovations in amyloid in the UK. There have been a number of trials trying to take advantage of this over the past 20 years. Prothena has invested in NEOD-001, looking at a number of different trials in AL amyloid. And you can see there have been some disappointing results. Most recently, a post hoc analysis, that is after the original trial, identified that people with really advanced AL amyloid cardiomyopathy may benefit from this antibody administration. There's Calum with 11-1F4 that is enrolling in a stage 3 AL amyloid cardiomyopathy trial. And then anti-SAP, which is Mark Pepys' primary project with GSK. The development has been discontinued. The drug that was developed successfully eliminated a ground substance stabilizer for amyloid deposits, but the amyloid deposits did not dissolve, even when an antibody against the deposits was added. And then finally, we have in Prothena the one TTR candidate, PRX004, which has been shown to be tolerable and safe, but we don't have extensive data. So the antibody story is very exciting, addressing not the production, but the dissolution of existing amyloid deposits. It just has not yielded the positive results that we had been anticipating over the past couple of decades. And finally, we have gene editing with CRISPR-Cas9 and Intellia's work. And in this graph that was presented at the International Liver Conference in June, looking at accelerating doses of this combination to the liver in a very small number of people. And I would emphasize, we're just looking at proof of concept here. We're not looking at clinical data. And what they measure is whether the drugs can scramble the TTR gene, preventing a message that can produce TTR from ever appearing in the liver. So the liver production of TTR drops rather significantly, and you can see it happens very quickly after the administration of drug. And at its greatest, nadir reaches 93% suppression, although my suspicion is that it's going to be somewhere between 87 and 93%. And ultimately, it's unclear whether a change from 83 to 85% suppression with the drugs that are repeatedly administered as gene silencers will differ clinically from gene editing, in which 85 to 93% suppression is achieved. That little difference in floating TTR around the circulatory system may or may not prove to have clinical impact. So we have a whole variety of things. It's sort of a luxury of riches here. Um, let's talk about the disease course and how to apply these drugs. So in TTR disease, whether we're talking about polyneuropathy or cardiomyopathy, there's a spectrum of disease that the audience knows exceedingly well. They're the experts. And in neuropathy, you go from walking unencumbered to requiring aids, ultimately to being wheelchair bound or bed bound, with progressive neurologic function occurring. And similarly in cardiomyopathy, the same process occurs. Over time, the compensatory mechanism of the organ that is primarily attacked, whether it's nerves or heart, those compensatory mechanisms diminish as that primary organ is more and more diseased, until you reach a point which I refer to as the tipping point. At that point, there are no compensatory mechanisms left over, and each small incremental damage to heart or nerve results in a dramatic and rapid decline clinically. So when you approach tipping point, you've got to have the best therapy available. So gene silencing therapy by whatever mechanism is really mandatory as you approach this tipping point. When you're earlier in disease, however, there are more options. You can preserve TTR presence by stabilizing that protein. So if you talk about paradigm, and in speaking in generalities for the BU group, our philosophy is in early stages of disease to use oral TTR tetramer stabilizers when possible. That if there's progressive disease, and if we're talking about neuropathy, to consider maximizing that tetramer stabilization by accelerating drug dose, as has been done with tafamidis, going from 20 to 80 milligrams, or with diflunisal going from 250 milligrams twice a day to 500 milligrams twice a day, monitoring renal function. Or to add or substitute with a TTR gene silencer. If you have multi-organ disease at the time you're presenting, then I would seriously consider early use of TTR gene silencers. And if you're a heart transplant recipient, that's not the end of the story, because the liver factory will continue to make abnormal protein, or in the case of wild-type TTR, a protein that can lay down on top of amyloid that has been deposited in other parts of the body, because we're talking about a systemic disease. So those people, post-transplant, require TTR treatment. And in the United States, orthotopic liver transplant really is outdated. And I can't remember the last time we referred to a transplant program with which we work. This is the amyloid group without whom I wouldn't be here. They make every day rather exciting, if not contentious. So it's really great to work with them. And with that, I will hold further comments and look forward to some questions.
Excellent. Thank you so much. That was a phenomenal talk, and remarkable to see the progress that liver transplant is no longer the only thing that you're offering for treatment.
Yeah, it's really true.
We have some great questions, so I'm going to dig right in. And one of the questions was around clinical trials. Does functionality override the typical other markers, such as scans or bio, when given a prognosis, so blood tests and labs and things like that?
Yeah, I think that's an interesting question, because from the perspective of the ultimate regulator, the FDA, they really want to know that someone's life is impacted. So if you show them modified neurologic impairment scores +7, they get dizzy. They don't know really what that means. They want to know that someone is better. So when we talk about the cardiomyopathy studies, the big advantage they have is that they've adopted the 6-minute walk test. But at the same time, um, that, um, that adoption needs to recognize that the ability to walk requires a heart that functions, lungs that function, and then legs that work. And in order for legs to work, you've got to have muscles and nerves. So just wild-type TTR, we know now, has neurologic manifestations, spinal stenosis, there's lots of radiculopathy, there's a peripheral neuropathy. And how much that's going to color the primary endpoint that you're talking about is really something that needs to be considered when you are taking a drug that you are convinced is really effective, but you have to prove its effect. And you have one shot. So if you take a measure that's going to be confounded by a bad back, you really jeopardize the drug program. So you have to be really careful. But I think it's an important point. Functionality is really the essence of what we're trying to achieve.
And with the follow-on to that, what's the level of difference that you would see with the walking test that would be considered clinically significant with the 6-minute walk?
Yeah, it's important. So 6-minute walk is now being shrunk to a 12-month period. And the concern I have about that is that we're talking about something that can be confounded by a bad knee, a bad back. Doesn't have to be just the heart. You know, your nervous function can change over time. And as well, a 12-month period is really just the beginning of the story in cardiomyopathy trials. All-cause mortality, you've got to be treated for 18 months. Cardiovascular hospitalizations, you have to be treated for 9 months before you get a benefit. So 12 months is really, I think, a little early to be measuring things. I understand the importance of an early answer, but the earlier you go, the less confident you can be that you're going to be able to demonstrate drug effect. So what is the difference? The experts will say something on the order of 15 to 30 meters at 12 months.
Thank you. So I have one more question in a line of tests, which is, how do you measure TTR levels? How's that test administered?
Yeah, so that's pretty simple. And you guys can all request the test from your primary care physician. It's called a pre-albumin test. It's a time-honored test for nutritional status, is the way we use it in the hospital. So every hospital has it. Every lab can do it. It's a really cheap test. And what it does is it gives you what we believe to be a biologic measure of drug effect. With TTR stabilizers, we're trying to increase the TTR levels, stabilizing it, preventing it from being lost to amyloid deposits outside the blood vessels. And in TTR gene silencers, we're obviously trying to stop its production. So, depending on the drug you're on, we either want it higher or completely absent.
Thank you. So there are quite a lot of questions about combination therapies.
Yeah.
So that might be something.
Yeah, no, I think that's really important. So
Yeah.
You've got two drugs, they work in different ways. Why not just use them together? Biologically, that makes complete sense. I'm not a big fan of it for one reason, except in one instance, which I'll get to. The reason I'm a little bit of a skeptic is TTR gene silencers are way more effective than protein stabilizers. And I think that's true with neuropathy and is going to prove true in cardiomyopathy. If the gene silencer is stopping production of protein effectively, there will be very little protein floating around the bloodstream. The target for tafamidis in cardiomyopathy is the protein floating around the bloodstream. If there is very little, then what is the function of tafamidis? The only way it can be additionally beneficial is if it has direct effects on the heart, independent of its stabilization of TTR, which effectively doesn't exist. Now, there's one caveat here, which is the obvious one. Your gene silencer is not completely stabilizing, is not completely stopping production of TTR. So you measure your prealbumin at home, and it's 7 to 15, and you're like, oh my God, this is fodder for progression of disease here. In that instance, adding a protein stabilizer, whether it's diflunisal or tafamidis, depending on a variety of issues, that to me is reasonable. And the other issue when you combine is you're talking about a drug that's $250,000 combined with a drug that's $500,000. And now you really are the $1 million person every year. And that's going to break the accounting for many countries. So I think we need to be a little bit thoughtful about this.
There are a couple of questions that are around people who are early in their disease and what's the best course of treatment. And even if you're asymptomatic, so asymptomatic or very early.
Yeah. So what we consider asymptomatic today is going to be the basis of therapy tomorrow. And what I mean simply is, at the moment, we rely on some indication of drug activity. People are born with a mutation, and we're looking for a sign of disease. What are the signs of disease? They're amyloid deposits or they're end organ deficits. And that means the process has been going on long enough for there to be some damage. Well, that's not the best approach. What we really would like is to know when the protein misfolding begins or overwhelms quality assurance mechanisms, when it becomes at a level that's a problem. And so for asymptomatic people, this is a huge question that pharma is going to address, and that is, do you simply start people on a protein stabilizer? The argument from companies that have TTR gene silencers is, well, if you look at the data, these drugs are really effective, but they can't turn back the clock really effectively. They can stop things with enthusiasm, but revising the damage, that's really difficult. So their argument is the earlier you start with one of these TTR gene silencers, the better you preserve what you have, and then presumably the better your quality of life going forward. TTR is a protein that's hanging around and has done so for millennia, and typically that signals that there's an important biologic function. What we understand of TTR is that it has secondary roles in the transport of thyroid hormone and a primary role in vitamin A distribution. But those are easily managed. It's not a critical element. There must be, I think, something else that we're missing so far. I tend to, therefore, be an enthusiast of sustaining TTR levels until that therapy is proven to be insufficient, and then to transition to something that's going to be more effective at truly stopping disease.
It really is a new frontier.
Yeah.
So there was a question that's saying, why does disease progress even after a liver transplant in some patients?
Yeah. So this is the flypaper phenomenon. That is that amyloid deposits that exist have the capacity to promote further amyloid formation. So you have a little spot of amyloid somewhere, and you have normal TTR floating by. it can actually stick to and add to that deposition disease. And this was first recognized about 5 or 6 years after liver transplants began in 1990. And we found at BU that by echo alone, it appeared that people who otherwise had successful TTR, successful liver transplants, were having progressive cardiomyopathy, infiltrative cardiomyopathy. And it was shown that wild-type TTR was developing essentially on top of the variant deposits that had been present at the time of the original liver transplant.
And we had a couple of questions about tolcapone, and do you want to talk a little bit about that?
Yeah, so yeah, there's a little bit of a pet project of mine. We're going through a renaissance here, a disease that was killing people at a very early and unfortunate age, or going through, declining cardiac function. But for the people with variant TTR, familial disease, their survival is being extended, hopefully to that of normal people. In so doing, we have to be sensitive to where we're controlling TTR production and where we're not. And where we're not is the brain and the eye. And the reason is simple. We can't get drug there with the drugs that we have. This is a dilemma that is in the crosshairs of 12 mutations that have early leptomeningeal disease. But one of those 12 is something called V30M, which is the most prevalent mutation in the world. And what I predict is going to happen, and I think most people involved in the game of amyloid agree, is that as we extend people's survival, we're going to start to see the inadequacies of our much improved therapies. We have to get drug to brain. Tolcapone is a drug that was designed for Parkinsonism. It is designed to cross the blood-brain barrier. We've demonstrated in a phase 1 study that it stabilizes TTR tetramer, much the way tafamidis and diflunisal does, both in the blood and in the fluid around the brain. We need drug to brain. That's simply stated. And Alnylam has entered a partnership and with the intent of developing drug for the brain and for the eye. So when you talk about the future of this disease, a lot of people focus on CRISPR-Cas9 and gene editing, which to me is sort of another form of gene editing, gene silencing that we already have. I think really the next innovation is going to be to get body-wide therapy, and that's going to include the brain and eye.
I think that maybe answers the question, and we are running out of time. So the last question, what excites you most about the future?
Yeah, to me, I think it's really about leptomeningeal disease, because while we're talking about only 12 mutations now with early onset, it's going to be everyone later. And the talk about Alzheimer's is deafening as you get older in life, and I think that's going to be the same thing as we happily see people with variant disease aging in a normal fashion on these really powerful TTR manipulating drugs.
Absolutely. So thank you so much. I do have, hold on a moment, I'm going to pull something up and then I'd like to thank you properly. First of all, there are a lot of questions came in. We will get to those and we will pick Dr. Berk's brain to make sure that we give the answers he would give. I also wanted to share something quickly, which let's hope I can find it. All right, let me just share this. So the Amyloidosis Forum is a partnership that's between ARC and the FDA. And very excitingly, we entered this data challenge that we're doing to really look at data across different trials and centers. And we entered it into an NIH challenge called Hero X, DataWorks Challenge. They have 5 cash prizes and 2 that are voted on by the public. So we need your help. And if you could go to archi.org vote, this is crowdsourcing. So every vote counts. If you have a phone, an iPhone, you can hold it up to this QR code on the right and scan it. And that will take you to the site too. But archi.org/vote. You do have to register to vote, but honestly, it would be fantastic if amyloidosis could win this challenge. We're in the short list and it's gone from 106 entries. We're in the final 20. So we would love your support on this. And while I leave this up, I would just like to take final moment. Dr. Berk, as always, it was a fabulous talk, just so clear and concise and thoughtful, and I think really understanding not just these endpoints that are used in clinical trials, but what the impact is on patients. And I think they can be very different. And so framing it that way is very helpful. So
the one thing I'd like to end with is that I would encourage everyone who's watching and has disease or has a family member with the disease to understand the disease, because ultimately, the therapeutic decisions are ones that you should make. It's your physicians who should lay out the possibilities, but you play a critical role in the choice. And with the development of multiple drugs, it's really critical you understand it. If they talk to you, doesn't make sense, stop them and tell them to put it in English, because you guys have the brain power and you have the commitment and interest. So really encourage you to know your biology.
And if you're not fortunate enough to be seen at an amyloidosis center and you don't meet regularly with a Dr. Berk or the equivalent, we also at ARC have numerous resources. We have a series of booklets. We also have a nurse specialist that's available to answer questions. So please feel free to reach out at any time. You can find us at arci.org. Thank you, Dr. Berk. We've really enjoyed today's webinar. Thank you all. Goodbye.